Okay, good morning, everyone. Today's open research office is going to be on TriNetX, and we're going to take a deeper dive than we've done in our previous session. And our speaker is still the same. It's Matthew Skowronek. So, Matt, when you're ready, you can go ahead.
Thanks, Marshall. And thanks again for having me. Everyone, like Marshall said, we're going to sort of take a deeper dive into TriNetX, looking a little bit more at the analytics that are run in the platform. So I'll do a really brief overview of our querying, our query builder. I know we've done that in the past, so I'll go over that quickly with some of the simple analytics that we have, and I'll sort of jump into our treatment pathways and compare cohorts, or compare outcomes, just to sort of give a little bit more depth. And then I'll also go into more depth about the process of requesting a data set to get row level data from the TriNetX platform, as well as the process for what we call export ID, which is running a query on the Buffalo network itself, and then being able to actually walk that the query results back to patient IDs. So you could use that for recruitment, feasibility, things like that. So I'll go into a bit of a slide on that. And then I'll also be able to show what actually a data set looks like if you download it, so you can be prepared for what the actual data you'll get. But we'll just jump right in to the platform. So just give me 1 second while I share my screen, everyone. All right, so some of you may recognize this query if you were on the last open research with me as well. This is the one I'll show, so I'll go over it very quickly of sort of making it, and then I'll jump into the analytics to show a bit more and get more insight into the analytics than just sort of glazing over like I did on the last one. So every time you jump into TriNetX, you'll actually be brought to your My Studies page, which will look like this. So it'll have every single study that you've ever created or that has been shared with you from other colleagues at Buffalo. To create a new study, you click on Create New Study, and there are 3 fields you actually have to fill out. So that would be study name, your research purpose, which for most, you can just do explore patient populations. And then down here at the bottom, you also have to add a therapeutic area that you're looking at. So once you do all that, you'll be able to click on Create, and it will bring you to your query builder page, which will look like this. It'll be a blank query builder. Up here on the top left, you'll see it says research. You'll actually be able to select whatever network you want to run your query on. So whether it be the research network, the Buffalo network, there are some other networks that are available as well. So you'll be able to hit down this caret here and select the network you want to look at. For this query, we're going to do it on the research network. So you can see within that network, we have 125 million patients, and there's 89 healthcare organizations on it. So we're just going to start out by naming our query, which is always helpful for the analytics, which I'll show after, but makes it a little less confusing when you're trying to bring in your cohorts. Oops. Just name it Buffalo there. So we're just going to do a quick Crohn's disease and ulcerative colitis just to do a brief overview of it. So typing into TriNetX is just like typing into Google. It uses predictive text as well as synonyms for it, so you don't need to know coding, the medical coding or anything like that. So we're looking for ulcerative colitis. We can see here it's K51, and we're able to just click on it. And then we're also going to add in Crohn's disease. So you can see here K50. One thing to note with the query builder is on the must-have side, it will always automatically go to and. So you'll make sure if it's an or instance, you'll want to just hover over it and click on or right there. On the cannot have side, we're going to say we don't want anyone in our cohort to have taken Humira, have a C-reactive protein level over 5, or have had an endoscopy in the last year. So we're just going to start out with Humira. You're going to say when we type it out, we'll see Humira is bolded down here, but it's also mapped to the generic. So anytime you query for a brand name medication, it'll automatically be mapped to that generic. If you're only interested in looking at that brand name medication, you can actually hover over it, click on this funnel that we have here, and you'll be able to select the specific brand name if you're interested in that. Also, if there was a specific route of administration or specific strength, you could do that there too. We're going to add in the C-reactive protein level here. And you can see there are a number of them here. You'll also see we have this TNX curated. So this is actually created by our informatics team. It includes a number of different codes. So instead of having to go through and click all the ones you like, this was actually created by our informatics team. So we're going to go ahead and select that one. And once you're in it, you can actually click on show included link codes, and it'll actually show you which codes are included. So if there were some missing that you wanted to go back and add, you could do that. But we want it to not be above 5, so we're just going to click on here, click on greater than or equal to, and then add in 5 and save that to our query here. You can see on the cannot have side, it automatically goes to or, so we're good on the or side there. And lastly, we're going to say we don't want to have had an endoscopy within the past year. So we're just going to click on there. It adds it to our cannot have side. But to add in that time constraint, we're going to click on create new group. If there were multiple terms you wanted to add, you could click on add terms and you'll be able to go and click the checkboxes for all of them. Or if you just have one like we have here, you can also just click and drag them down. For the time constraint, we're just going to click on time constraint here. If you had specific start and end dates, you would be able to add those here. So you can see specific start and end dates. Because we are only doing a blanket 1 year, we're able to use this ruler bar here and add in 1 year. You're also able to get more granular if you want days, weeks, or months in there as well. So we're going to go ahead and save that. And then we're just going to also adjust a demographic for this. So we're going to look at only patients over the age of 18. So where it says any age, any sex, we're just going to click on this caret here, and we're going to do greater than 18. If you are interested, you could also filter by males or females for your query. For this, we're going to do both, and then we're just going to save that criteria again, and then count our patients here. So you'll see from our query, 87 of the 89 healthcare organizations returned results for about 436,000 patients on it. To take a bit of a closer look into some of the patients that are in this cohort, we're going to use this Explore Cohort button here. Just click on Run, and this will give us a breakdown of the patients within our cohort. So we'll be able to look at the diagnoses, procedures, meds, and labs that all of these patients also have within their record. And then the percentage of patients within this cohort that have those as well. So you'll be able to see here, if it ever gets caught up a little bit on this 97, it normally takes less than a minute to come in, but you can also look here, stop and view partial results if it does get hung up in the 90s there, and it will eventually show all the results for you. Within the demographics, you can just see the breakdown here. So we have the breakdown, the males and females, also have ethnicity and race. You'll see our min age is 18 as we set, but the max age will always be 90 in these. You'll see the number of patients over the age of 90 listed here as well. And then we can jump in. Diagnoses, procedures, and medications are all formatted exactly the same, so I'll just show one. So we're looking at diagnoses. So say if we wanted to just look at some diseases of the respiratory system, we can just use these carets on the side here. We just look at some influenza and pneumonia. We could see that 8% of our patient population within our cohort also have had pneumonia in the past as well. You can get as granular as you want with these using all of these. And if you were looking for specific covariates, you could search them in here as well, either by the name or if you knew the coding as well, you could search for that. Procedures and meds are formatted exactly the same, so I'll just skip over those and we'll jump into labs. So these are grouped into lab groups. So we have the metabolic panel, CBC, liver function. We go all the way to the bottom here. We have all of our vitals. And then right above that, we have this area called findings. So findings is anything that we gather from the healthcare organizations that also provide notes. So we have a natural language processing vendor that goes through those notes and pulls out extra information that's not structured within the EHR. So you can see here we have ECOG scores, left ventricular ejection fracture, and depending on the query that you run, you'll see more and more items within that findings tab there. Below this, we have analyze criteria. So the analyze criteria is sort of looking at the impact that each query item has on the actual end result of your query. So we're able to select our baseline. We'll do the UC and Crohn's. And then we're able to select the terms for analysis. You can do up to 15 of them. We only have 2 extra there, so we're just going to click on run. But it will show us a funnel graph from the most impactful item within our query to the least impactful. And you can use that as sort of a hypothetical. This is always a bad excuse because there's such a large number of patients that come back for it. But say if we didn't care if our patient results were being a bit limited, we could say, oh, maybe if we remove the fact that they couldn't have had Humira, we would be able to just click on hide here. You can actually see the number of patients that would be added to your cohort if you did remove that Humira there. And this is all hypothetical, so you can add them right back. If we didn't care if they had endoscopy, you could add that and see a 5% increase in it. But if you did see something limiting your cohort that didn't completely affect the question you were trying to look at, you'd be able to go back into Query Builder and change that out there. One thing we also have within Analyze criteria, if you click on this View button here, you're also able to see the breakdown between races, sexes, races, and ethnicity. We call this our diversity lens. So say if we just wanted to look at sexes here, you could see the breakdown of which is more impactful between males or females, or in this case, there's a unknown as well. So you could see those. We're able to do that all within the Analyze Criteria tab. Rate of arrival, I always show on this. This is one of the simple analytic tools that would be run just if you were running the query on your own HCO network, because you can use this for feasibility. So it uses 3 years of historical data within the cohort to predict the next 12 months of data. So predicting the next 12 months of patients that would come in that directly fit your cohort. If we click on show graph here, we could see that 11 months ago, there were 3,800 patients that came in that met the criteria perfectly. And then moving forward, you could go out as well. So 8 months, there'd be 3,700. Really good for feasibility. And this could tie into using the export ID button as well. So just to jump into that, the export ID button, it's actually a really simple tool to use within the platform. You would click on these 3 dots on the right side of count patients, and you would actually be able to just click on export patient IDs, which I have grayed out on my screen here, but you would have it available on yours. And once you click on that, it will go to the honest broker that is at Buffalo. They'll make sure that you have the appropriate IRB in place and everything for it. And then they would be able to actually run the export patient ID. All the patient IDs within the TriNetX platform are shifted from what's actually in the EHR. So they have a crosswalk back to it. And the team with Jonathan Blazier and Viraj, they would be able to crosswalk that back to the actual MRNs within within your EHR, and you'll be able to get those patients for recruitment. At the end, I'll show a quick slide on sort of the process, and then I'll make the 2 slides I have available as well after. So if you are interested more in the process of that. We also have summary statistics, which if we just run that quick, and the joys of doing a live demo is you get kicked out of the platform. But summary statistics, where Explode Cohort, you were looking at sort of the patient data itself and the patients behind the data. The summary stats, you're actually looking more at the facts and the actual data of what would be included in a data set download if you were to perform one. So it gives you the total number of patient facts that you would have, the average number of facts per patient, and then sort of breaks that down into the length of the patient record, the fact distribution, what those exact fact types are. So diagnosis, procedures, med labs, but also um, genomic data, oncology data, or chemotherapy data if there's available. And then within the platform, it also gives you a geographic breakdown. So it breaks it down into 4 regions of the US, also ex-US, because there are some non-US healthcare organizations on the research network. And then other regions or the unknowns are all located within the US. They're just community-based hospitals, so it's easier to jumble them together instead of pinpointing each and every one throughout the US that we have there. In a dataset download, you'll get a little bit more information on geography as well. It will break it down depending on the size of the cohort down to the census regions. So it'll be broken down a bit more than just Northeast, Midwest, South, and West that we have here. And as we're talking about data sets, you're able to request data sets just on the platform like this, both from the Buffalo network as well as the other networks in TriNetX that you have. So the Link network and the research network that we have. And that can be done simply by clicking this request dataset button, the big yellow button on the right here. And then you would just select which query you wanted. You can select multiple queries. You'll see that some of them are grayed out, and those ones are normally grayed out because they're running on a network that does not allow dataset downloads. Or they include terms that you can't download in a dataset. So there are sensitive terms within the platform that you're not allowed to run analytics on or get data sets for. But you would just continue, you would click on request here. We would go through this data set process here, and then you would just type in a quick 1 sentence of what your study is actually going for. If you're interested, if it was a very large cohort and you would rather have a random sample of it, you could click on the random sample and add the number of patients, and then you click send to TriNetX. In that process, this is for the research network. It would go to Jonathan's team, and then they would send it over to me. I would send a data set order form over to them that would be signed for a signature. It's more of a terms of use. There's no cost for any of these research level data sets unless it's being used for grant funded research. And then we would just generate it through the platform, and all of the data sets would then be available in this available data sets tab here on the left. I have a slide for this as well, and I'll show that afterwards. It has the sort of the workflow of it. But one more thing I wanted to show, or 2 more things I wanted to show that are just in the platform here. We have this study management tab. So like I said at the beginning, when you're in your My Studies page, it'll show you all the studies that you've created, but also studies that have been shared with you from other colleagues. So within this team on the side here, you can actually share your studies. These are all TriNetX members, but say if I wanted to share it with Aaron, I could write the reason why I'm sharing it with him and then click on Save Team. It would actually add the study to his My Studies page and also send him an email notification that a study was shared with him. He would then be able to go into the study and work on anything within the query builder that we have here. All of the queries that are run are all date and time stamped, and then they're also stamped with the individual who ran the queries. So if there are multiple users on one study that you're working on, you could be able to see the work that they've all been doing. And you don't have to worry about accidentally deleting things. It's sort of a 2-step process for it. So if you were to click on delete, you would have to click on continue as well. So it's not an accidental deletion thing you would have to worry about. The last thing in the platform that I always like to show, which is super helpful for all of our investigators at our HCOs, is what we call design assistance. So this is a direct link to our trial design and optimization team. All the analysts on that team monitor these. You can just type in any question that you have with your query. Maybe something is really limiting your results, or you're just having trouble finding certain codes for something. Or you just want an extra set of eyes before you go and request a data set or something like that. You can just type in your question there, click on submit question, and you'll get a response back from our TDO team. It's normally within 1 business day, and it'll start an email chain between the analyst and yourself. So if you do have additional questions, you don't have to go back into the platform and ask them on this screen. You can just keep replying to that email. So you'll know you'll be working with that same analyst for all of that time. And then just to jump into some of the analytics. So we do have a number of analytics on the platform. I'll show 2 in depth today, but I'll show you all the ones you should have access to here. Today, I'll show you the compare outcomes as well as the treatment pathways. There are very, very good training documents on all of the other analytics as well within our training center, which I'll show at the end as well. Specifically, there's webinars specifically looking at the advanced analytics as well. One specifically goes into the incidence and prevalence, which is one of our newer analytics. So you'll be able to see that, and it's done directly from our trial design and optimization team. But for this, we're going to show the treatment pathways of this. And this is why I said it's really important to name your queries. As you can see on the left side here, I have a lot of names, but for every analytic that you run, you just click and drag over the query you're looking for. So we're going to do a previous one that I already have built out. But you're looking at the IBD demo here. You will click on go to characteristics. So you'll go to characteristics here. You're actually able to set your time window. So this is any time before, up until 1 day before the event. And in this event, it's the diagnosis of Crohn's or ulcerative colitis. You can see that here that we selected that as our index event. Once you have just those 2 things selected, we click on run, and it'll actually populate all of our demographics, diagnoses, procedures, meds, and labs again. But within this, you can also star anything you would like. If you would like to use those as your treatments at the end, you can see here we saved a couple of the treatments that we have that will show up in the end, and you'll see them. We run this query again, or this analytic again. This time you're looking at the time after the event. So this could be any treatment after the event of the original diagnoses. We're doing an anytime ending on this, so there's not a hard stop. You'll see on our compare outcomes, we institute a hard stop on that. But it's exactly the same. You just add in your index event, which is the first diagnoses, and then we click on run, and it will just show us the procedures and medications that we have here. So you see if we jump to procedures, we have a number of procedures here, and then just as we were doing before, you can get more granular in these that we have here. And you'll see anything that is blue is actually an item within your query. So we have endoscopy here, and you'll see that there as well. And within these, we actually went through and saved a number of different treatments that we had. After you go through that, we'll jump down to the input section. So this is where you'll select those treatments. If you didn't go and star any of them in the original 2 steps, this is where you'll add in those treatments that you want to look at. Again, just with that time window, exactly the same as the last one. But as we scroll down here a little bit, we're able to add in our treatments. If you did star any of those treatments, they'll be listed here in this drop-down menu. You can just add them all to it, or you can use this search bar just as we are doing our query, and type in either the name, the code that you're looking for, or anything like that to add them to the query here. You can also add a grouping. So we use the example of if you're looking at different chemotherapies, and there are a number of different medications that go into it, you can add a group for it, and it'll actually be named the group instead of showing each medication individually. So you're able to do that just by clicking on this create group here. But once you have all your treatments added, we click on run, and it will generate our lines of treatments that we have. So we're able to look at the treatments within the pathway, the distribution of treatments across all the lines of treatments, and then it'll show us a sunburst diagram of those different lines of treatments. within this first one in treatment pathways, it'll show the top 10 treatments within this, which is within this query that we selected. If you click on select treatments, it'll actually give you the entire list of treatments that we have. So if you were interested in something else besides the top 10, you can actually uncheck it and check what you are looking for. But in this treatments, if you see here and you click or hover over anything with those interlocking circles, you can see that there are 2 treatments. So it's a combination treatment that we have there. And if we move over to the right side, I always say sort of ignore the adrenal corticosteroids. It's the clear #1 course of treatment for all of it. But below you can see sort of the distribution of which would be the first line of treatment, second line of treatment, third line of treatment, and so on. And then as we jump down here to our sunburst diagram, we can see just how that goes. So 46% of patients use adrenal corticosteroids as their first line of treatment. And then say if we wanted to jump out, 2.97 went from adrenal corticosteroids to omeprazole, and then back to adrenal corticosteroids, back to omeprazole. And then once you get to other is anything outside of those top 10 treatments that we had in that first visual that I showed. But the good thing with all of our analytics is you're actually able to click on this download button here. You're actually able to download the image files and the data all behind the imaging. So you'll be able to get more info on what that other is. I believe it was 0.4% followed that specific group, but you'll be able to get all of that data here. You can do that all individually with the downloads if you're looking for each individual graph, but you can also click on export all, where you'd be able to click and choose which you wanted from everything within that specific analytic. We also have compare outcomes, which I'm going to show a different outcome here for a different study that was created by our clinical or trial design and optimization team. They changed names. They used to be our clinical sciences team. So if I intertwine them, it's a little messed up in my head a little bit. But it was looking at the administration of warfarin and apixaban and then some outcomes with it. So we're going to look at outcomes of major bleeding, outcomes of stroke, and a lab value for these as well. But with all analytics, you start with just clicking and dragging over your patient cohorts that you've created. So there's one created for warfarin and one created for apixaban here. Next, we would go to characteristics where we're able to set our timeline before. So this is any time before up until 1 day. And you can see here we've created our apixaban and warfarin cohorts, and then we were selected them as our index events that we have here. You can see these are much larger cohorts as well. And I'll get into some of that on how our design assistants can be really helpful within this process here. But once we do this, we're able to click on run, and we're able to get a breakdown of our demographics. So it'll give us a breakdown of our demographics, just as we had with our other analytics and just as we had in our Explore cohort. So let's just jump into it here. So we have the breakdown of our demographics here. We have current age, age of index, sex, ethnicity. We're also able to see the difference between the 2 cohorts that we're looking at here. And within these, you can see that some of them are starred here as well. And if we jump down to baseline comparison statistics, anything that we starred within our characteristics tab, we're actually able to see here. So if there are number of values associated with them, you're able to get a little bit more information on these stats. So we have the mean, the min max. You'll have the p-value and the standard difference for all of these characteristics in here as well. And then with these characteristics that we've chosen, we can also use these as covariates when we are doing propensity score matching. So the next step here is the balanced cohorts. All of these are actually the ones that we've previously selected from our cohorts. If you were looking for, again, something specific to add in that you didn't find to star, you could just add it in your add covariates section here, just as we are adding to our query builder. but you're able to select all your covariates that you want to look at, and then you're able to click on run, and it would actually bring up our propensity score matching. So we can see our propensity score curves that we have here before and after matching. It'll show us all of our characteristics as well that we selected before and the after matching. And then we're also able to look at the outcomes. So the outcomes we want to look at here, we're going to jump. You're able to select if you wanted to do this compare outcomes before matching or after matching. And then you're able to set your time window. So you can see here, this is where we have a more finite time window than the previous one. We're looking at from 1 day after the administration of apixaban or warfarin up until 3 years after that administration. We're just going to scroll down past the query that we have here and then jump into our outcomes. So we're able to select all of our outcomes, which are easily done. I'm just going to jump to the bottom just by clicking on new outcome that we have here. And it'll again give us a search bar that we have here. If you did have any outcomes saved, you could click on them there and add them individually. For this one, we added them all just through the search bar. So with our first outcome, we're looking at stroke. So we added a number of stroke terms here. And then you're also able to select what analytic you want to do along with this. So we're going to look at a measures of association as well as a Kaplan-Meier analysis for this. And then our next outcome, we're going to be looking at major bleeding. This is where I think our design assistants can come really helpful when you're looking at certain outcomes. With major bleeding, there's such a vast number of terms. And on our TDO team, they're all experts in finding all of these and working with the platform to get the most out of your cohort. So you can see just by the sheer number of them, I think there are 30 plus codes in this one to look at. But again, we'll just be looking at a measures of association for that. And then our next is we're just going to look at an INR value. So we're going to grab our INR there, and we're going to look at the lab results. So we'll be able to see the distribution curve of that. Once we have all of our outcomes selected, we're just going to go ahead and click on run. We'll actually be able to see all of our results. And we're going to run that. Let's try. Again, the joys of doing a live demo when it doesn't, keeps kicking you out. All right, but we'll be able to see all of our measures of association as well as our Kaplan-Meier analyses that we have for this here. So if we just jump into outcomes here and the results, all of these results for the compare outcomes will start with the propensity score matching. So what we saw in that first part of the balanced cohorts will show us again here. Be able to just fly through those. And then we'll jump into our first outcome, which was a stroke. And we'll look at our measures of association. So it includes all of our cohort stats, risk difference, risk ratio, and odds ratios. And then our Kaplan-Meier analysis. Again, our cohort stats has the hazard ratio as well as proportionality. And you're able to see the curve a little bit better if you expand it like this as well. Again, like we did with treatment pathways, you're able to download all of these individually if you would like, or use this export all button to click and choose which ones you would actually prefer. We'll jump back down to major bleeding where we did, again, measures of association. And then our INR just to show the lab distribution that we have here as well. One thing I'd like to note with all of our analytics that we have is if you did run an analytic and like you saw previously, it was saying it wouldn't return results. Maybe if there's an HCO that is going for maintenance, all their data is under maintenance, sometimes you won't return the same results as you were thinking you would get. So in this history tab here, you can actually go back and go to previous results. So if you were running it and for some reason your results weren't coming up, you could go back and actually click on a previously ran result and get those analytics that you have here. So it'll give you the date and times of when they were run. and you could just click on it and go right back to it. And it just keeps a running log of every time you click a run button, it'll create a new history for this. All right. So within this demo, I talked a little bit about data sets as well as the export ID function. So I just want to show those processes a little bit. I have a few slides that I want to show for this. All right, so if we were to look at this one here, so this is the export ID process that we have here. We would be able to request the patients for ID. So that would be actually those three little dots to the right of count patients. And you would be able to click on count patients. That would then go to the honest broker at Buffalo. They would be able to determine if you had your IRB. An obvious one there. If you didn't have it, you could submit it to use this export ID within your process. If you did have it, you could easily go click that export ID button. The IDs would be generated. It's delivered in a password protected Excel file to the user that selected it. So it would send it to them. They would actually be able to go back and match it to the MRNs within your EHR. In turn, you could then create your patient list for your study team, and then go through the whole pre-screen process and everything like that. So the export ID process within the platform is really easy. The main, I would say, work on that side is on the Buffalo side after you get the actual de-identified list of patient IDs and they map them back. So it is a little bit more extra work on the Buffalo side for that. But that is that process that we have there. We also have the dataset workflow. So I kind of went over this a little bit just to show it a little bit more in depth here. So within the TriNetX platform, you're able to create your query. We always try and recommend to use design assistants before requesting a data set. Just asking them, hey, does everything look good? Does this answer the question that I'm trying to answer? Just so you don't have to keep requesting data sets if you find out the data isn't included that you actually need. The user will be able to request a data set, which would then be sent to Jonathan Blazier and his team. And they would be able to send that over to give the approval and then send it over to TriNetX. I would then get that data set request. I would be able to send over the data set order form. If it was for a non-funded study, there would be no cost included with it. If it was for a funded, grant-funded data set, it's an added step here that's not included. So it would be between the HPM sends a data set order form and the HCO returns the signature. We would just jump on a really quick phone call, talk about the study that you're running, the size of the study, so the number of patients that are in the cohorts, and then the size of the grant that you're looking for. And then we would come up with a price for the data. We would get the signature back from Jonathan, and with the Buffalo ones, we have a signature. It's a 2 signatures, so Jonathan would sign, and then the PI investigator would sign as well. As soon as I return or get that back, I would start the generation process. Normally, it's done within a day, the generation. The paperwork part is normally the part that takes a little bit longer. But on the platform, depending on the size of it, it could take a couple hours to probably a full day to generate, just depending on the size of the actual cohort that it is. But once it is complete, I go back into the platform and release it to the user who requested it, and it would be available in that available data sets that I showed earlier on that left-hand side there. And when you download that data set, it would come to you in multiple CSV files. So they're all merged in this one, but let's see here. Everything you see here on the bottom, so there would be a patient CSV and an encounter, diagnoses, medications, procedures. They would all come in separate CSV files that you would merge together. But you can see it includes a patient ID. This patient ID is also one step away from the synthetic patient IDs that are within the platform. So there's actually no way to go back from a data set to re-identify patients, even if you ran it on your own Buffalo network. So just something to keep in mind. Data sets are completely de-identified even on the TriNetX side, so there's no way to crosswalk that back to use expert ID afterwards. You could go back and use expert ID in the platform, but you wouldn't be able to sort of compare that and join that together with a data set. With all data sets also come a data set dictionary, the most recent data set dictionary for whatever network you're on. So it'll give you a breakdown of everything that's included in the data set, what some of these things might mean. So if you're looking at diagnosis indicator derived by TriNetX, anything like that, it goes into more depth on the tumor registry and tumor properties data as well. So if you're doing an onco study, there's a lot more details within that data set dictionary that will all be included in there as well. So that was that. I just want to show a few more things on the platform before jump off here. But within the platform, we also have our help and training center. So you see here, if we click on, it would be to the left of your initials, is this question mark here. And we jump into the help center. We'll be able to see our training center, our FAQs, and our resources tabs. Within our training center, it has a list of webinars that have been completed by our TDO team. It's a pretty expansive list that we have here. So we'll show here, like I talked about, there are specific ones on the analytics as well. So if you click on TriNetX 401, it gives you sort of a brief look at what you're going to see in them, but it's broken down into two different videos as well. So they're about a half hour long for each of them, but they're broken down into treatment pathways and incidence and prevalence, as well as compare cohorts, outcomes, and analyze and compare outcomes. Easier done than just sort of cramming everything within one webinar. If you're looking for one specific thing, you can go right into those and find those here. They also have one with competing risk. So if you're looking to use competing risk, they have those there as well. Also included in these we have a analytics strategy guide. So you're able to look at the strategy guide that we have here. And then it shows any help center articles that might be of use to you as well. So I always use the incidence and prevalence because there's not a demo for it that I normally do. But there is a really good help center article on incidence and prevalence. So you could see that there. And then we could actually go back and show how that would be shown on here as well. One thing I'd like to point out is within this TriNetX training program site here, it also lists the upcoming live training webinars. So our TDO team holds 5 training webinars per month. 2 are sort of the basic, the query building, and then the basic analytics, which are everything on that left-hand side. So the explore cohort, summary stats, and things like that. There's also 2 advanced analytics trainings where they get into more depth of incidence and prevalence, treatment pathways, things like that. But there's one rotating one as well. So for April, it was best practices in querying oncology. And for May, they're actually doing their first best practices in querying pediatrics. They haven't done one of those yet. So that'll be May 16th. All of these can be registered for right here on this webpage as well. But some others that have been part of that rotating one are the querying oncology, querying rare disease, and then querying diversity were also big ones that have come up in the last few months that we have had here. Within here as well, this was the training center. The FAQs is where you'll find some of those help articles that were linked within the training center. So if we scroll down here, we have incidence and prevalence. has 8 actual full articles on incidence and prevalence. They have this on all of our analytics as well as the networks that we have available. So if were interested in certain networks and you didn't really know what they were, like the TriNetX Link network, which is our network that also combines outside closed claims data and extra mortality data within the network, you can answer all of your questions on here as well. And then the last thing I wanted to show that we have here on our sort of public facing TriNetX page, so just trinetx.com, not the actual platform webpage, we have a full, under the resources tab, a full publications page. So you're actually able to see all the most recent publications that have been done with TriNetX. You might also see some press releases come out through LinkedIn about some of these. This most recent one was looking at long COVID with the FDA. So you're able to see that. And then you're able to see these as well. So they're actually all broke down. You're able to look at different tags for them. So if you're looking at or looking for different ones with autoimmune disease or pediatrics or psychiatric, just to see the level of publications that are being done through the platform from our HCO members and the journals that they're being published in. You can see all that here. We also have our data privacy attestation. So if you're looking to see a little bit more into that, and we also have our publication guidelines, which gives you a little snippets that you can throw into your methods section if you are using TriNetX within your citations. So you're able to see here, very easy, just sort of copy and paste that in so you don't have to do all that as well. It also gives you some guidelines on if you want to use logos for within your publications or within posters or things like that. All that can be found here within the publications guidelines. All right, I think that concludes what I have here. Alexis, Marshall, did you have anything you wanted me to add in here?
I don't. Alexis, did you?
Nothing really for you to add, except I'm going to share a screen in case any of our attendees have not registered for their own account on TriNetX. This is the contact information for Garage, and so you can do the registration that way. But then if anyone has any questions for you, Matt, I guess that we can open it up there.
Yep, absolutely.
I don't see any questions in the chat. Does anyone want to unmute themselves and have a question? I don't see any hands raised.
And if there are questions that come up afterwards, maybe if you haven't been in the platform before or in a while, I'll send over these the screenshots of the slide deck and also provide my email so Alexis and Marshall can distribute that as well, so you can ask questions directly if interested.
Okay, well, no one has any questions. So, Matt, thank you for presenting again today. And everyone else, thank you for attending.
Yep, thanks everyone.